So what we need with hypoimmune, and I put here from one of the publications, a modified figure where we really worked with islet cells. We are really interested in the type 1 diabetes space. So it was an islet cell. And when you transplant that from a donor to a recipient, you get immune rejection, and we knocked out the beta-2M and C2TA. So we removed the MHC molecules on the cell surface of those cells. And that we do because they act as the fingerprint. So when you remove the fingerprint, the adaptive immune cells, the T cells, they don't see that you transplanted a cell that has a different fingerprint. What I also like about knocking it out and not just knocking it down is that those MHC molecules, they are presenting neoantigens, MIHA, so minor histocompatibility antigens. And when you knock them out, you don't have that presentation to the immune system at all. But because then the innate immune cells, macrophages and NK cells, they are in our body to sense what we call a missing self. So that is a cell without fingerprint. They would attack immediately. And what we did in the past is that we overexpressed a molecule that was called or is called CD47. And that can inhibit innate immune cells that they don't kill when they are triggered through a missing self. So that work has been published. We will not go into those published data because we want to focus on xenotransplantation, but I thought it makes sense to highlight the last publication, and that was with a PI, Professor Carlson. He is the principal investigator at the Uppsala University, and what was done is hypoimmune engineered islet cells. So they didn't have the MHC molecules, they did have the CD47 overexpression, and they were transplanted into the forearm of a 42-year-old patient. You see that here, the transplantation itself. And the patient was type 1 diabetic since his childhood. And he had no measurable C-peptide. That was an inclusion criteria. And what you see on the graph on the lower here, on the lower row, is that before the transplantation, there was no measurable C-peptide. And then the x-axis shows you the days after the transplantation. The publication reports up to 3 months, but you might have seen also now the 6 months data presented by Professor Carlson. And you see that there is measurable C-peptide. C-peptide levels are really low, but that was on purpose in that study because the dose was so low. Patient didn't have any response to a mixed meal. So you see at baseline here, C-peptide levels wouldn't go up, but he responded after the hypoimmune transplantation. So that is a nice proof of concept that you can use genetic engineering, modify cells, transplant them without any immunosuppression. The patient didn't get any immunosuppression, nothing that would alter the immune system, no steroids, etc. It was pure injection of the hypoimmune islet cells.
Long story short, the first successful swine kidney that was transplanted in March 2024, and a second case done recently at Mass General, and 2 other great cases done at NYU. The patient at NYU actually did very well for up to 4 months before requiring dialysis and explantation for a number of reasons of really rejection. But again, it's a proof of concept. Really, really exciting work. Heart and lung transplants have also been done recently at NYU in Maryland, and a lung was recently done in China, which is the patient's doing quite well by all reports. So the resources that have been put into this field now are really finally coming to fruition. There was a big pause in xenotransplantation for many years with what I consider unfounded concerns about retroviruses that turned out to really not be very important or meaningful. And so now we're off to the races. Clinical trials are now funded and ongoing at MGH and NYU, and we're looking forward to this new age, golden age of xenotransplantation. However, there are still problems to solve with this. Can you advance the slide for me? Thank you so much. So before we talk about that, I just want to tell you, so when I joined David Sachs' lab, he had gal knockout swine and was using the skin from these swine to help soldiers. That was his idea, to put them on to burn patients and have them be temporary skin coverage, just as we use aloe skin for coverage. Well, now we routinely use this product about a decade later in wounds such as this. This is a sweet, healthy, otherwise healthy little old lady who lost the skin on her leg from an infection, and they wanted to amputate this leg, which is basically the mortality of when you had an amputation at 86 years old, let you stay in bed, you get diabetes and bed sores, and your mortality is about 60% over 2 years. So we used a gal knockout swine skin. If you want, I can advance here. In the OR, this is a approved product by the FDA now, where we're able to take these with no donor sites now and place it on her leg. Next slide. In the OR, and you can see this incredible. They come in quite small pieces at this point, but they heal quite well. And this is just creating a wound bed. I won't say that this stays on and leads to this result. We do have to go back and autograft with the patient's own skin over this, but it covers tendons, bones, exposed nerves, things that you couldn't cover with just a skin graft. And we're able to save this woman's leg. And it was really a remarkable clinical result. And if you look at the next slide, we've done this now in a few patients, and you can see the results that, even secondary healing is better than a normal allograft with this technology. So exciting proof of concept, and not quite a kidney or an organ or heart or lungs, but our humble plastic surgery contributions are still important and still can matter to patients. So.
And you do see here the data. So we had the human endothelial cells, and when I write here HIP with that star, what that means is they have the hypoimmune edits, so the MHC class 1, class 2 knockout, and then the star means the expression of the host CD47. So in that case, it was a macaque CD47 overexpressed, and then they were transplanted into a macaque monkey, so xeno, crossing the xeno barrier. And what we learned is when we did the blood draw after the transplantation in the monkeys, for the T cells here, that the T cells were not activated because we had knocked out the MHC molecules of the transplanted cell. And how you read those graphs is, so you see here the percentage drop of the cell index at 90 hours. And the cell index itself shouldn't drop. So we are doing basically over 90 hours a killing assay on our platform where we can measure if the cells are surviving or if they get killed by the effector cells. In that case, we added the T cells. And what you see is in gray, before the transplantation, nothing happens. The immune system hasn't seen the transplanted cells. But 7 days later, you see here in blue, also the macaque T cells are not recognizing the transplanted cells, and that is because of the MHC knockout. So you want to have that here at 0. And then you see for the macaques NK cells as well, when we overexpress the macaque CD47, there is no activation. The NK cells are not killing the missing cells. Same for the macrophages, and same was true when we run an ADCC with NK cells or macrophages. So we really were able to address that innate immune killing in a xenotransplant model by just using the host CD47. However, we run into severe issues with another innate immune cell population, and that were the PMNs. So polymorphonuclear cells are usually not recognized in publications. If you look at detailed immune analysis, you will hardly find any team, any group that studied the PMNs. And that is okay, most likely for the allogenic field. However, in xenotransplantation, we learned that it is a major issue, because what the PMNs are doing, they get activated mostly through the toll-like receptors, the 4, 7, 8, 9, and they get activated by DAMP signaling, by xenoantigens, and by cytokines. So this is a huge issue because those cells, they kill immediately. They are like an innate immune killing. They don't even have to learn. They get activated, they kill. And what you see here is that even before the transplantation, the immune system doesn't need to learn about the transplanted cells. They're killing immediately. And they did so also at the day 7 after the transplantation. So we had to find a way to tackle the PMNs, which is one additional layer of complexity if you want to go into hypoimmune for xenotransplantation. With cells, when you do the genetic engineering, that always has some risk with it. It's really a risk-benefit question. But for the xenotransplant field, I think we can think about organs, etc., that will be safe. And there are the concerns, and more work needs to be done, no question. But We also know now that a beta 2M knockout cell, etc., can still clear viruses. There are enough publications now out in that field.
Yeah, I would just also mention the concerns about porcine endoretrovirus or PERV have been made a big deal of for a very long time. We actually there are many background swine that has PERV knockouts, but even whether that's necessary is even in question. We have an FDA approval for use the skin that we use. For example, we had safety concerns. We did many assays at many times and found no PERV transmission between the swine skin and the host. And if you go back to the original work on that, it's very difficult, even in vitro, for this virus to infect human cells. So it was something that's I believe it's been accepted as a real risk-benefit ratio, as Dr. Schrepfer said. So I don't think I think there's certainly other infectious diseases in any transplantation we should be aware of, and safety assays are obviously critical, but I think the benefit of the organ being able to be transplanted is far outweigh these risks that have turned out to be minimal to none. It's actually quite remarkable how little downside there is as far as infectious transmission of these porcine concerns so far.
Great. Here's another question from Dinashari Survey. How long did the transplant cells stay? What is your perception on the risk of tumor formation using the genetically edited hypoimmune cells?
So to the first question, the study I showed you with the PMN inhibitors, the 5-drug regimen, was really hard to do in the monkeys. It is so toxic. So if you look in the publication, we pulled it after 2 weeks. So we had certain times in the paper, but nothing of that can be done long term. So we're planning now with Professor Cetrulo together studies where we're now really looking on pig cells with human and do the transplants there. So